Oh no! My bio project is due tomorrow and I haven't even started!
We can help you with that!
What? Who are you guys?
We're the Bio Wizards! And we're gonna help you with your project. What's the subject on?
Well, it's an in-depth analysis on mitosis and meiosis.
That sounds boring. Yes, he's right, but we can still help you with that anyway. Let's start with mitosis. What exactly is mitosis?
It's easy. Mitosis is a cell division that results in two cells having the same number of chromosomes as the parent cells. And meiosis...
Exactly right. Yes, you are. But what about the detail, like the phases and the cell components involved?
Cell components are centrosomes, chromatin, nuclear envelope, nucleolus, centromere, chromosomes, and the kinetochore.
Now let's talk about how those cell components work in mitosis.
First of all, the nucleolus is something that contains genetic information in a cell, and the nuclear envelope surrounds it, and as mitosis goes on, the nuclear envelope disappears, and the genetic information that was contained inside the nucleolus gets condensed into something called chromosomes, and those are held together by centromeres.
Mention that not only is the nucleolus inside of the nuclear envelope, but there's also something called chromatin, which is just the unraveled form of chromosomes. And organelles called centrosomes release thin, fibrous, stringy things called spindle fibers. And the spindle fibers attach to something called the center of the chromosome, called the kinetochore.
Joe, you forgot to mention that the centrosomes go to the opposite sides of the cell as they form the spindle fibers.
I don't care! Now let's see what you know about the phases of mitosis, Alexey.
Wow, I actually know this stuff. The first phase the cell goes through is interphase, which does not involve any dividing. The first part of the interphase is the G sub-0 phase. This is when cells do not divide at all. Some of these cells include neurons, which rarely divide. The cell then goes through G sub-1 phase, and this determines if the cell will divide. The S phase then replicates the DNA and synthesizes proteins. The G2 phase then also synthesizes proteins and begin to start the mitotic spindles in their earliest stages.
Is that all you know?
Yeah, dude.
Well, since you're dumb, I'm going to explain to you what happens after the phase, which is the mitotic phase.
The first part of the mitotic phase is prophase, in which the chromosomes become visible and condense, spindle fiber forms, and the nuclear envelope dissipates.
Prophase, which Joe wonderfully explained, comes the phase prometaphase. And what happens there is anything that's left of the nuclear membrane completely dissipates, and the spindles start to actually attach to the chromosomes. And after prometaphase comes metaphase, where the chromosomes start aligning in the middle at something called the metaphase plate.
Good job, Rounak. Next comes anaphase, in which the chromosomes split into sister chromatids and move to either side of the cell near the centrosomes. Anaphase comes telophase, in which the cell cleavage furrow becomes visible and you can see the cell separating, as well as the nuclear envelope forming. After that comes cytokinesis.
Ooh, I know this one! This kinesis, the cytoplasm splits, creating two daughter cells that have identical genetic information.
Wait, Alexey, don't forget that each daughter cell has the same amount of chromosomes as the parent. But in humans, such cells have 2 copies of 23 chromosomes called diploids.
Thanks, guys. Now let's look at a brief overview of what I've learned so far. As we go through interphase, the amount of DNA stays at 2n. As we approach S phase, the amount of DNA duplicates to 4n. And as we go through the mitotic phase, through prophase, metaphase, and anaphase, the amount of DNA still stays at 4n. As we approach telophase and cytokinesis, since the cell divides, it would go back to 2n as it approaches interphase again. I've been studying inside for all day. I think I need to take a break and go outside.
Alright, Alexey, now that we're done talking about mitosis, let's talk about meiosis.
Great idea! So, Alexey, what do you know about meiosis?
Absolutely nothing.
Looks like AP Bio did a lot for you. Anyways, let's start with the basics. Now, there are 2 parts to meiosis, meiosis 1 and meiosis 2.
These 2 and meiosis 2 are virtually identical. They both consist of prophase, metaphase, anaphase, telophase, and cytokinesis, which is very similar to mitosis.
In fact, the only difference really is that the full chromosomes are attached instead of the sister chromatids.
Right. For Meiosis 1, we're going to name the phases Prophase 1, Metaphase 1, Anaphase 1, Telophase 1, and Cytokinesis.
Same goes for Meiosis 2. The phases will be called Prophase 2, Metaphase 2, Anaphase 2, and Telophase 2.
Phase 1 of meiosis, the nuclear envelope begins to break, and the centromere begin to make spindle fibers. As well, the chromosomes will do something called crossing over, in which they share genetic information with each other.
Metaphase of meiosis 1 is where the tetrads are attached at the kinetochore by spindle fibers, and they begin to align at the metaphase plate.
Anaphase 1 of meiosis is when the individual chromosomes are pulled apart to each side of the cell.
It's getting a little dark out, so I think I'll start studying inside too.
Now, after anaphase 1 comes telophase 1. This is where the cleavage furrow, thanks for reminding me, cleavage furrow becomes visible and the nuclear envelope forms.
So then cytokinesis must be where the cell splits into the two normal cells again.
That's correct.
Awesome! So what happens in meiosis 2?
There are many similarities between meiosis 1 and 2.
That's very true. The phases are all the same, except that instead of tetrads, they are homologous chromosomes.
That's easy. So then the processes must be the same.
Prophase 2 forms spindle fibers, and the nuclear envelope breaks apart.
That is true. And also, metaphase 2 lines up the chromosomes at the metaphase plate and spins out the spindle fibers.
So then anaphase 2 must split the chromosomes, and telophase 2 and cytokinesis must split the cytoplasm in the cell.
But unlike in meiosis 1, where they become 2 end cells, in meiosis 2 they become 4 individual end cells.
Now, this is called a haploid cell, but another name for that is a gamete.
Thanks, guys. This has helped so much. I think I'm going to finish this project now.
Wait, Alexey!
We forgot to mention other applications of the cell cycle.
Like apoptosis and cancer.
Wait, how do we get outside?
First, let's talk about cancer.
Cancer is a disease in which the cells uncontrollably divide.
So, it's when the cells have cell cycles that are corrupted.
So basically, cleavage furrow.
Wait, what?
Right, Alexey, now let's show you a demonstration. No, I'm uncontrollably dividing.
We're cancerous cells!
I'm here to bully you!
Nerd! You freaking nerd! You nerd! Get down, nerd!
Talk about apoptosis.
Now, it's simple. The concept is simple. All that really is just programmed cell death.
Hey, can I see a demonstration of that?
Sure! Okay, Alexey, how about we show you a better demonstration? Say we have a fellow cancerous cell right here, and since it's harming the body, the brain has to shut it down somehow. So, it has to undergo apoptosis. Die, cancerous cell!
Oh no, I'm undergoing cell death.
Allahu Akbar! Alright, kids, let's go over a brief synopsis of what we just learned. First, we talked about mitosis.
Then, we talked about meiosis.
And finally, we talked about applications of the cell cycle, such as apoptosis
And cancer.
Hello class, my name is Mr. Gopal Shater and welcome to AP Bio class hour 1, 2. Okay class, I want to see your homework right now.
Oh gosh, I've worked on this bio project for so long. I hope it goes
I don't
well.
care, give it to me.
Okay.
Okay, let's see here. AP Bio project cleavage burrow. That's very good. That's very good. I don't see your name on it. You get C minus.
What? No! My parents are gonna kill me.
Okay, class, it's Mr. Gopal Shader, AP Bio hour 3, 4. I mean, 1, 2. Okay, 1, 2. Okay, class! I need your homework right now.
I have my extra credit, sir.
What is this?
Okay. So,
Hold up!
Take your time.
Okay, what is this?
Okay. So I did my extra credit on cancer. This is obviously when the cell cycle abnormally divides, and we have a tumor here. There are two types of different tumors. There is a malignant tumor, which is more severe,
And then there's a benign tumor, which is not as severe.
What is difference? One kills, one does?
Yes, the malignant
very
tumor
good, very
kills.
good, very good.
And we also over here have apoptosis. This is a cell before it's undergoing normal cell division. It's a normal cell, and after it's a destroyed cell.
What happened here? Alakbar?
Yes.
Okay.
Do you think it's good?
Yeah. Okay, now face the wall.
Okay.
Face. Okay. You get C minus.